NURUL WAHIDA BINTI ABDULLAH, and you guys can call me WAHIDA.
Hi everyone, I'm NABIHAH.
Assalamualaikum warahmatullahi wabarakatuh. My name is Wan AZMIRUL IZRAN bin Wan Muhammad MIRZA.
Hi everyone, I'm LOOSHINIE KUMARAVELU.
Hello everyone, I'm Muhammad MIRZA MIKHAIL. Today our group, the Susis, will be talking about our product, the sausage. As a host today, I would like to brief a little bit about our product, the sausage. The sausage that we buy in store are mostly raw, and then we cook in many different ways, such as pan frying, boiling, or barbecue. But did you guys know about how the process of making the sausage? Did you know? Most people think that they just stuff the meat and shape it into cylinder shape, but that is wrong, Sausage makers salted various types of tissues and organs such as scraps, organ meat, blood, and fat to help preserve them. After that, they stuff the tissues and organs into tubular caning of animal intestine. Is clean, okay? And producing cylinder shape that we are called now the sausage. Sausage is a type of meat product usually made from ground meat, often as beef, pork, or poultry, along with salted, spice, and other flavorings. Next, I would like to talk about the history of sausage. The word of sausage was first used in English was around in 15th century. That is spelled like this. This word came from the Old North French that is called saucisse. Did you guys know that sausage is actually one of the oldest preserved food? This is because some research has found an Acadian cuneiform tablet. Acadian is East Semitic language. There is a record, a dish of intestine that is stuffed with some sort of force meat. In China, around 500 BC, there is a sausage called lukjiong. Lukjiong is made from goat meat along with onion. Well, every country has different type of sausage, And lastly, there is a famous sausage in Italy that is called Lucanica. Lucanica is from Lucania, that in modern society nowadays called Basilicata. Well, I think that is all from me. Now I'm going to pass to our next presenter, LOOSHINIE.
Hello everyone, now let us look at the principle of microorganisms and product. Did you know that sausages are made with the aid of bacterial fermentation? Well, bacterial fermentation is a process in which microbes convert these sugars in food into other compounds such as acids, gases, and alcohols, which gives the meat its unique flavor. This sort of sausage is often fermented with bacteria found naturally in the raw meat or artificial starter cultures added to the raw meat during the production process. It has long been recognized that starter and protecting cultures are critical in the production of safe and high-quality fermented sausages, and lactic acid bacteria such as Lactobacillus, Streptococcus, Pediococcus, Leucolostoc, Lactococcus, and Enterococcus, as well as Staphylococcus, are the major factors in the production of fermented sugars. These organisms perform three functions simultaneously in fermented sausages. First, they produce nitric acid by reducing nitrate and nitrite, which is responsible for the cured color when combined with myoglobin, and they lower the pH by converting glucose to D-lactic acid via anaerobic glycolysis. LABs, which is lactic acid bacteria's primary function is to convert fermentable sugars in sausage batter to lactic acid, which helps to keep the food safe by creating unfavorable conditions for pathogens and spoilage organisms. Lactic acid fermentation is influenced by time, temperature, and relative humidity. When sausage is fermented at the right temperature and facultative anaerobic conditions, the LAB grows rapidly, converting simple sugars to lactic acid and lowering the pH. When the sausage are smoked, the smoke can contribute thousands of aromas and antimicrobial compounds such as organic acids, and also contributes to the coagulation of surface proteins and inhibition of microorganisms. The breakdown of flavor and aroma producing compounds like myofibrillar and sarcoplasmic proteins is done with the help of Lactobacillus plantarum and Lactobacillus casei. The lactic acid bacteria used in the production of fermented sausages cannot be chosen randomly. Hence, there are several criteria that need to be followed for a good quality of fermented sausage to be produced. The selection criteria are fast production of lactic acid, good growth at different temperatures, homofermentative metabolism, persistence over the whole fermentation and ripening process, nitrate reduction, ability to express catalase, no fermentation of lactose, formation of flavor, no formation of peroxide, no formation of biogenic amines, no formation of ropey slime, tolerance or even synergy to other microbial compounds of the starter culture, antagonism against pathogens, antagonism against technologically undesirable microorganisms, improvement of the nutritional value of the sausage, and economic factors. All of these factors must be taken into account before selecting the suitable starter cultures to be used during the fermentation process of the sausage. That's all from me. Thank you.
Thank you for your explanation, LOOSHINIE. Moving on, now I would like to have NABIHAH to tell us about how the process of producing our product.
Hi everyone, I'm NABIHAH and I will be explaining about how to make a product which is sausage. There are several processes of making sausage, which are selecting ingredients, grinding meat ingredients, blending meat ingredients and non-meat ingredients, smoking and packaging. First and foremost, meat needs to be cleaned to prevent contamination with bacteria. Selecting a correct amount of spice and seasonings also important to create satisfactory product to stabilize mixture and adding flavor to end products. Size grinding stage is needed to reduce meat ingredients to small uniformly sized to create sausage with protein and small protein and fat content. This stage is varied according to production and nature of the product. Moreover, blending of meat and non-meat ingredients are required for specific sausage formulation and also to obtain uniform distribution within product wording and consistently mix in entire sausage formulation. Furthermore, sausage use smoking. Smoking is used to dry and cure meat for making smoked sausage and disseminate aroma to final product. It is important to reduce bacterial growth in the finished product. Automatic smoking method is used to produce healthy meat. Last but not least is packaging process. Product may be wrapped in gas-resistant plastic and placed in refrigerator, display and follow sanitary way to avoid infection of product. Normally, many sausage are vacuum packaged to maintain freshness and avoiding them from freezing. Method used is to eliminate air from package and wrap paper in zipper bag of freeze items for 1 to 2 months without affecting the quality and taste of the sauce.
So that's how the process. Oh, and by the way, thank you Nabiha for your explanation. Next, I would like to have AZMIRUL to tell us about the application of product.
There are many types of sausages that are made with the aid of bacterial fermentation, such as the salmon sausage and the dry-aged sausage. The process is also known as spontaneous fermentation. The process involves the addition of the lactic acid bacteria such as Lactobacillus, Pediococcus, and Streptococcus. These bacteria will produce lactic acid that will convert sugar in the sausage into acids, gases, and alcohol. It also gives a unique flavor to the sausage and make the meat become dry and to avoid the spoiling of the sausage.
Thank you AZMIRUL. Now I would like to have our last presenter, WAHIDA, to share us about the benefits of our product.
Today, I'm going to explain about the advantages of using bacteria in producing the dry sausage. So, lactic acid bacteria has long been utilized as a starter culture in producing the dry fermented sausage or any other meat-derived commodities. This culture is designed to meet the virtual safety, shelf life, technological efficacy, and economic feasibility criteria. Biopreservation refers to the elongated storage life and enhance the safety of the food by utilizing the natural microflora and their own antibacterial product. Meanwhile, the lactic acid bacteria has a major potential for use in biopreservation because they are safe to consume and they can naturally dominate the microflora in many foods. In milk, brine vegetable, or any foods with integrated carbohydrate, the lactic acid bacteria is used to develop the food products. In raw meat or fish that are chilled under the vacuum or in environment with ascending carbon dioxide concentration, the lactic acid bacteria will become the ascendant population that preserve the meat with a hidden fermentation. The same applies to processed meat provided that the lactic acid bacteria can survive the heat treatment or they are inoculated onto the product after the heat treatment. Beside all these traditional properties, novel starter culture should take into account the perils posed by the formation of biogenic amines in pabulum, the development and spreading of bacterial resistance to antibiotic. Further, functional starter could protect the consumer from deleterious bacteria either by an expeditious acidification or by the production of antimicrobial. Some specially culled culture may additionally provide a probiotic benefits, and if appropriately modified, they may even be endorsed with nutraceutical trait. What is nutraceutical trait? It means that the food has a health-promoting benefit. It also has been proved in the research that fish sausage fermented by the potential probiotic of Enterococcus species from dry fish had valuable health-promoting benefit compared with non-fermented fish sausage.
Thank you WAHIDA. I'm so sorry to tell you guys that we have reached the end. I hope that everyone can enjoy and also can understand about our show today. Well, see you guys soon. Bye bye.